Hi everyone, we're going to pick up with the nervous system. In your course pack, this begins on page 92. Let's first start by talking about an organization of the nervous system. We have both a structural and a functional setup for the nervous system that we'll discuss. Of course, there's the brain, spinal cord, there are nerves called cranial nerves that branch off the brain, and there are nerves called spinal nerves that branch off the spinal cord. Now, when we talk about functional subdivisions, we're talking about how these portions of the nervous system operate. In other words, are they sensory in nature or are they motor in nature? When we talk about structural subdivisions, we're talking about where those parts of the nervous system are located. Brain and spinal cord, for instance, are in the central nervous system, while spinal nerves and cranial nerves form the peripheral nervous system. We'll talk further in later chapters about the branches somatic and autonomic nervous system and the parasympathetic and sympathetic nervous systems. But you'll notice that those mostly fall into a motor output or a functional category for our nervous system. Here you can see the central nervous system and the peripheral nervous system. Those are our two structural categories. The central nervous system includes the brain and the spinal cord. The peripheral nervous system includes nerves that branch off your brain and spinal cord. That includes cranial nerves that mostly serve your face, head, and neck area, and the spinal nerves that branch off the spinal cord. So when we're talking about central and peripheral nervous systems, we're really talking about a structural classification here, not a functional classification. So let's fill in this box. It's found on page 92 in your course pack near the bottom of the page. I am going to mark out these bottom two boxes here because they are not relevant for the central nervous system. So we've discussed that the central nervous system has the brain and the spinal cord, while the peripheral nervous system includes cranial nerves and spinal nerves. So these are structural categories that, in other words, helps us describe the structure and the way this nervous system is set up. Functionally, the central nervous system serves as your control center. It not only receives incoming sensory information, it also relays outgoing motor information. So, the central nervous system makes decisions based on incoming sensory information, and then it dictates the outcome. It dictates motor responses based on that information that comes to it. All the peripheral nervous system does is act as a transporter, transporting sensory information to the central nervous system or relaying motor information away from the central nervous system. The peripheral nervous system does not function as a control center. So we either send sensory to the CNS or receive motor information from the CNS. There are functional subdivisions of the peripheral nervous system. You may recall from that previous slide, we looked at things like the somatic and autonomic nervous systems. And of course, those are both motor in nature. Okay, so we also have sensory information that comes to the brain and spinal cord via nerves that travel over those peripheral spinal nerves and cranial nerves. So when we're talking about functional categories, we're talking about motor output, sensory input. Okay, so the sensory portions of our spinal nerves and cranial nerves take information to the CNS. And those are receptors that we have in glands, organs, and muscles. And like we just mentioned, the motor output comes from the CNS. And that includes the somatic nervous system, which is voluntarily controlled, and the autonomic one, which is involuntarily controlled. Here you can see a summary of the functions of the nervous system. Sensory information is carried to both brain and spinal cord over spinal or cranial nerves. The brain and spinal cord make decisions, and the motor output then is carried back out from the central nervous system over motor pathways to skeletal muscles or glands or organs. And those muscles, glands, and organs carry out the responsibility of doing whatever it is the central nervous system dictates. In fact, we have neurons that perform specific functions. We have sensory neurons that carry information to the brain and spinal cord, and we have motor neurons that carry information away. So you can see in this picture, the sensory neuron takes information received from receptors. These receptors are shown here in the skin. Those sensory neurons carry information to an interneuron, which you can see pictured here. And this interneuron actually communicates between the sensory neuron and the outgoing motor neuron. That tells me this sensory neuron is outside the CNS, while the interneuron must be within the CNS. And of course, the motor neuron is outside the CNS as well because it carries information to a skeletal muscle, gland, or organ. So if you'll take a moment and flip on over to page 93 with me, you'll notice that these bits of information are for us to fill in the top of the page. Sensory input has receptors in glands and organs. The integration process occurs in interneurons in the central nervous system. And then, of course, we stimulate motor output by taking information to effectors such as muscles, glands, or organs. You've only got 2 blanks there, but you can jot down those 3. We have effectors in muscles, glands, and organs. In this image, you can see a sensory neuron here taking information to the central nervous system. Of course, this is the spinal cord. The spinal cord right here is part of the central nervous system. Inside the spinal cord, we have interneurons. Here they are. And they're the ones that are communicating and making decisions, receiving information from sensory neurons, and dictating information to these motor neurons that carry information back out. So the only decision-making part of our entire nervous system is our brain and spinal cord in the central nervous system. So the next portion of your worksheet has a blank. Interneurons are found only within the central nervous system. Sensory neurons are outside the central nervous system in the PNS, or peripheral nervous system. Motor neurons are actually cell bodies rooted in that central nervous system, but they continue on and become part of the PNS as well. So sensory neurons may be located inside your body as interoreceptors. They may be located outside to pick up parts of the outside world, such as taste receptors. They're called exteroreceptors. And then we also have proprioceptors, which are found in joints. That gives us information about the location of your body parts in space, even if you can't see where they're located. Let's talk for a moment about neuroglial cells versus neurons. We know that nervous tissue is composed of both types of cells. While neurons conduct nerve impulses, neuroglial cells do not. Neurons lose the ability to divide over time. Therefore, they cannot repair themselves if damaged. While neuroglia cells can actually repair themselves, they never lose the ability to divide. Neurons are insulated, protected, and supported by neuroglial cells, and neuroglial cells simply are support. They do not have the capability to produce a nerve impulse. They are only there to help nourish, protect, support, and insulate those neurons. So we're going to spend a few moments talking about the types of neuroglial cells on the very bottom of page 93. If you have trouble getting this to stick, I recommend flashcards. Okay, so some of these are located only in the central nervous system. They include oligodendrocytes, astrocytes, microglia, and ependymal cells. Keep in mind the S words. Satellite cells and Schwann cells are only found in the peripheral nervous system as neuroglia. So let's first talk about neuroglial cells in the peripheral nervous system. Microglia are spider-like phagocytes. Phagocytes are cells that eat. So they are here to help clean up debris in the central nervous system. That means in the brain and spinal cord. And you can see in the image here, it really does take on a spider-like appearance. Astrocytes, by the way their name is implied here, are actually star-shaped cells. They help to brace the neurons and most importantly form a blood-brain barrier inside the brain. That blood-brain barrier is going to be a topic for our discussion a little later on. When we hear of someone having brain cancer, it's not the neurons that cause cancer. It can be cells like these astrocytes that can grow out of control and cause things like glioblastomas. Oligodendrocytes produce a myelin sheath around nerve fibers in the central nervous system. It is their job to help insulate and speed nerve impulse transmissions when we see those oligodendrocytes present. When you've heard of white matter or gray matter in the brain, it is the oligodendrocytes that form the white matter. The myelin sheath does give off a whitish colored appearance inside the brain. We're going to learn later on that the myelin sheath helps to speed up nerve impulse transmissions because it allows for nerve impulses to jump and skip along the length of the axon. Ependymal cells are also found in the brain and spinal cord. They line the cavities of the brain and spinal cord to help circulate cerebrospinal fluid. We're going to call cerebrospinal fluid CSF for short, so I just want to go ahead and put that out there. CSF circulates both inside and outside of your brain and spinal cord. These ependymal cells have cilia on their surfaces that actually help to create a flow for that fluid. So even if you're sitting or laying perfectly still, that fluid will still help to circulate. Now, keep in mind the two S words here are found only in the peripheral nervous system. Satellite cells and Schwann cells are in the PNS. The other terms are found in the central nervous system. We have satellite cells that help to protect neuron cell bodies. You can see one in the picture below. It's the Schwann cells that we will spend a little more time on. They form a myelin sheath just like oligodendrocytes, but they're called a different thing because of their location. Oligodendrocytes are found in the central nervous system, whereas Schwann cells are found in the peripheral nervous system. Now, both oligodendrocytes and Schwann cells do form myelin sheaths. The only difference is where they're located. Again, myelin sheaths form a white covering on the outer surface of axons. They do help speed up nerve impulse transmission, and they are there to help insulate and protect those very precious axons because, if damaged, those axons cannot be repaired. So nerve impulses actually go faster because they hop, skip, and jump from one of these little nodes to the next, right around the length of those myelin sheaths formed by the Schwann cells. We'll talk more about those on the next page in your course pack, that is page 94. Make sure you fill in the blank on the bottom of 93. Schwann cells produce the myelin sheath. Let's talk about neuron structure for just a moment. We know that neurons conduct nerve impulses, whereas neuroglia do not. Let's start by looking at the cell body. The cell body is the metabolic center for your cell. That cell body actually houses the nucleus and the organelles of that neuron. So the cell body here is the metabolic center of that cell. It has axon and dendrites that connect to it. So let's focus next on the axon. The axon is typically one single fiber that leads away from the cell body. You can see in this image we do have only one axon down here. It is connected by way of an axon hillock right through here. And the axon carries information away from the cell body. Okay, so that's important, that the nerve impulse only travels in one direction here. It always travels from the cell body along the axon away from that neuron control center. Alright, so the axon is usually one per neuron. They can be 3 to 4 feet in length. In the case of your leg, you do have some that travel that long down the length of your leg. We've talked about the axon hillock. Let's now talk about the other end. These axon terminals are bud-like endings on the opposite end away from your cell body. Those axon terminals will release neurotransmitter onto a neighboring cell. It could be a neighboring neuron, a muscle cell, or some other gland or organ. So those neurotransmitters will be the topic of our discussion when we discuss nerve impulse transmission. So the function of the axon is to take a nerve impulse away from the cell body and to create a synapse with a neighboring neuron or another type of cell. The chemical that the axon terminals release is called a neurotransmitter. That is the blank there on your worksheet. These dendrites are the regions that bring in nerve impulses toward the cell body. They're like satellites looking for incoming impulses. So the nerve impulse always travels in one direction only. That is from dendrites to the cell body, from the cell body toward the axon and away, and that of course terminates there with those axon terminals. Let's now focus on the myelin sheath. Okay, so just a look here at the axon. These have no myelin sheath. Here you can see a myelin sheath or a wrapping around the outside of that axon. And this is how a myelin sheath is formed. If you could imagine taking a toilet paper or, say, a paper towel roll, unwrapping it, and then wrapping it up yourself, you'll notice how you're wrapping around an inner cardboard core. That would be like the axon. So this cell wraps itself around and around the outside of the axon so that it creates a protective outer covering. And because it is a fattish white protein, it gives the appearance of white matter. So we do have white matter in both the central nervous system and the peripheral nervous system. When we're talking Schwann cells, which are we discussing? CNS or PNS? Schwann cells imply peripheral nervous system. Okay, so on your worksheet, the blank there is Schwann cells form the myelin sheaths that are found in the peripheral nervous system. They do wrap around the axon. There are gaps, however, between neighboring myelin sheaths, and those are called nodes of Ranvier. You can see the nodes of Ranvier here. It is a gap or a blank space between neighboring myelin sheaths, and that node of Ranvier actually allows for us to have an increased speed of nerve impulse transmission. So instead of the nerve impulse having to depolarize the length of the entire axon, it can hop, skip, and jump from one node of Ranvier to the next. That's what makes nerve impulse faster in white matter than in gray matter. We call that saltatory conduction because we're looking at a speeded up or an increased speed for that nerve impulse transmission. Finally, on page 94, you'll notice white matter versus gray matter. White matter is any case in which you have a myelinated nerve, whether it's central or peripheral nervous system, and gray matter includes those unmyelinated cell bodies, axons, and dendrites. Here you can see myelinated versus unmyelinated axons. The myelinated one has a myelin sheath covering the outer surface, and it allows for a nerve impulse to only need to depolarize along the length of those nodes of Ranvier. In an unmyelinated axon, we don't need to depolarize along every node because we have to depolarize the entire length, meaning that that nerve impulse transmission is just a slight bit slower. Just a side note here about MS or multiple sclerosis. In this case, we see that there are damages done to those myelin sheaths. In an earlier chapter, we talked about scar tissue. We know that scar tissue forms when there's damage done to cells, and collagen fibers replace that damage. So here you can see some areas of damage. Unfortunately, scar tissue will replace this damaged area, and no longer can that nerve impulse jump so cleanly from one node of Ranvier to the next. So in multiple sclerosis, we tend to see nerve impulses that go awry. Perhaps they are slower than normal, and so there may be areas of numbness or loss of muscle control where there has been nerve damage. In the top left picture, you can see a healthy nerve and how the nerve impulse hops, skips, and jumps from one node of Ranvier to the next. That is how it should work in a myelinated axon. But in the bottom right picture, you'll notice a damaged nerve with damaged or scarred myelin, and that nerve impulse does not really make it fully to its destination. And that's how we end up getting, say, like I mentioned, numb areas, parts of your body that don't receive the nerve impulse fully, so we have muscle weakness, for instance, that may result. Multiple sclerosis can also result from damage in the brain to the white matter. You can see the picture on the right shows plaques that result from scar tissue developing where the myelin sheath has been damaged. And here you can see on the right, again, myelin sheaths that have been damaged and the plaques or scarring that builds up in the brain. So this may result in difficulty with being able to see, smell, for instance, taste. Whatever part of the brain that may be damaged with this scar tissue results in perhaps some sensory or motor output problems.